Hey everyone, welcome back to the Genomics Bootcamp. This time it will be a bit of an unconventional video with a bit of unconventional presentation. The title is Diversity of Approaches to Develop Small Breeds from Community-Based Breeding Programs to Gene Editing. Originally, I gave this talk at a roundtable organized by colleagues from Humboldt University to Berlin, but then I thought it might be also interest to some of you. Before I start, a little explanation for those of you who are not really into livestock breeding. So in animal breeding, we often refer to small breeds, but this is not related to their physical size or height or anything, but rather than the population size. So there are large populations or global populations, if you will. For example, the Holstein cattle is always a good example that has millions of individuals all around the world. And when I say millions, I mean many, many million animals of the same breed. On the other hand, there are also so-called small breeds that are present maybe in just some country or even on a regional level. So it might be that there are just a few hundred individuals alive at any given point for particular breeds. So these are the so-called small breeds, and these are of particular interest if you are into genetics of diversity, and also if you want to protect these small breeds somehow. Now, this workshop I was attending, this was focusing on discussions around small breeds, and then, well, I also had this contribution in there. But as I mentioned, this presentation will be somewhat unusual because, well, I will be having a lot of questions and not necessarily the answers. But I also believe that asking the questions is also worth it. And also, perhaps it can trigger some discussions or at least thinking about the subject. So let's see where these questions lead us. So without further ado, the idea behind this presentation is to discuss approaches to improve local small breeds. As you will see, this presentation will be a bit of a roller coaster, talking about approaches business as usual, all the way up to similarities to 90s science fiction movies, or at least one particular one, which will be at the end. You will see. I will be asking a lot of questions, which can be summarized as, where are we? How can we do better? And what can we expect in the future? So let's start with the easier one and try to determine where are we currently with genomics in livestock. So right now, genomics in livestock based on single nucleotide polymorphism data, but also whole genome sequences is really current practice. There are a lot of applications and a lot of methods that can be used to improve livestock breeds. There is an excellent paper by George et al. From 2019, harnessing genomic information for livestock improvement. If you have the opportunity, I really suggest you to read it. But anyway, so at the conclusions, they state, among others, that the animal breeding just completed a prototypical once-in-a-lifetime Gartner hype cycle. So what is a Gartner hype cycle? I will explain in a moment, but I just want to underline that I question a little bit this once-in-a-lifetime claim that they have in this sentence. And that's because I think we might be entering a different hype cycle as we speak. Okay, but what is this Gartner hype cycle? So I pulled this one from the Wikipedia. It's actually some kind of a generalization that explains behavior. So on the x-axis, we have a time, and then on the y-axis, the visibility or kind of a enthusiasm of people about some issues. So there is a technology trigger at some point, and the hype goes up, and then there is a peak of expectations when we think that we can solve everything with this new technology. Unfortunately, it tends to turn out that is not so true, so there is a disillusionment. But then after some time comes the enlightenment and the plateau of productivity. Now, this could be really nicely translated and applied also for genomics. So in the 1980s, there was the emergence of genomics, and in 1990s, we had the quantitative trait loci and the marker-assisted selection, and we thought it will solve all of our problems. Well, as you might know, this was not entirely the case. We found out that not enough variation is explained, and we were generally sad about it. But then with the dawn of the new millennium, there came the whole genome sequences and the SNP chips, and we could cost effectively genotype a lot of individuals. That led to the boom of the business. And since 2010 and further up, we have the genomic selection and other methods that we use happily ever after, or so. So now it is a good time in livestock genomics, and we achieve some kind of a plateau of productivity according to this hype cycle. But of course, then there is a question, are all the breeds, all the regions equal? And the answer is sadly that they are not. In Europe, as far as I can tell, we are quite fine. Things are going reasonably well. There is recording for production, for pedigrees. There is a reasonable amount of genotyping going on. The records of various kinds are being used to explore the breeds and increase our knowledge about them. Also, of course, for their improvement in large breeds, these most populous breeds, or those which are used for production. Of course, the productivity is the main focus, not the exclusive focus, but still the main one. And in small breeds, in small populations, we tend to focus in keeping them alive and keeping their populations reasonably diverse. But do we have the same conditions elsewhere? Yeah, I would say yes. In North America, Australia, New Zealand, well, there are some countries that are so-so, depending on which region you look at. So in China, Brazil, India, perhaps some other countries that could be on this line. But there are many other regions where the conditions just totally not the same. So we have a lot of gaps that could be filled. We know that there are different challenges, and one of the solutions for these challenges are the so-called community-based breeding programs. They are an alternative to conventional breeding programs as we see them, for example, in Europe. And they can be applied even if there are some difficult conditions. For example, if the recording is not present at the project start, or in general, the breeding programs needs to be set up in a region where it was not done before. The main focus of these community-based breeding programs is the strong involvement of local farmers, and they were proven to increase the production of individuals or the livestock breeds, and also improve the livelihood of farmers. I have talked about community-based breeding programs in detail on this channel, so if you are interested, just look up my video about animal breeding in low-income countries. For this presentation, just a short summary. So typically what we see when we start with the community-based breeding program somewhere, that there is a local population of a local breed that, just generally is not so good in terms of production. Here are the picture from cattle, but of course, this is just an example, and the community-based breeding programs could be applied actually in any species, but the logic is always the same. So what we see all the time, so that the populations themselves are quite variable. There are individuals that do not have a very good production, there are some average ones, and there are individuals that are much more productive than the others. But let's say that the trait we are measuring is growth, so the size of the animals. But for an unmanaged population, it is really hard to identify the really good animals. And then what's tend to happen is that the population stagnate, or even it could happen that they get progressively worse. This is where the breeding programs come in, and because, the name is community-based breeding programs, the local communities and local farmers are also heavily involved from the beginning. The farmers state their preferences, and with the help of the research team, they categorize the individuals or animals into three groups, usually three groups, ones that are, not so good, some average, and those which are really, really productive. Then there is a committee that actually selects the parents of the next generation based on the pre-selection groups. Technically, they could select any individual as the parents of the next generation, well, usually the male parents, but they tend to select from the highest productivity group, sometimes if there are some specific requirements also from the middle group. But at the end, what happens is that the next generation gets slightly better. And if this process is consistently repeated, we achieve the increase of productivity even under very difficult conditions. This is a picture from such a selection event. This time it is a sheep breeding program in Ethiopia. After this short overview of the current situation, I want to follow up with thinking how we can do even better. Now I realize this is a really complex question, and I am focusing only on the genomics aspect here. And it's not that I am ignoring all these other aspects, but I just chose the genomics as the focus of this presentation. So that's why I'm going down this path. So the key question here is, what is our level of satisfaction with the current state of management in small breeds? Let's say when it comes to genomics. Well, the current status is that the genomic data, let's say SNP data, is really affordable. So I was thinking, why don't we have a genomic repository that has genotypes for everything? And if I say everything here, I really mean every single breed from every single species. Yes, we have a lot of possibilities. Yes, we have a lot of open access datasets, but I doubt that we have everything. Actually, I do realize that I have really high expectations here, but otherwise it would not be a challenge. What I have in mind is some kind of a resource or some kind of a place or repository or database or anything of that nature where we could go. And if we think about any breed whatsoever, we could pull out at least 10 genotypes or more for that particular breed, and we can do, well, something with it, or research or whatever we need it for. Apart from being completionist or some kind of data hoarder, I think it would have also some real advantages, and I will talk about these just in a bit. So what is the status on availability of genotypes and sequences, perhaps also in anonymized form? For large breeds, well, the more popular breeds of any species almost, I think we are quite fine. For smaller breeds, maybe not so much. Partly, yes, there are, in some cases, these things are available, but I'm convinced that there is still a lot of ground to cover. Now, there is this fairly famous saying, at least in livestock circles, that in the age of genotyping, phenotypes are king. But really, I want to focus on the first part of this saying. This first part actually implies that getting genotypes is easy. And essentially, it is true, it's easy. It's just really a function of money. And with all the research projects around, getting genotypes and also getting new genotypes is really feasible. So my repeated question again, why don't we get a repository of all livestock species of all breeds from Europe, from outside Europe? Yes, it costs money. It probably costs a lot of money, but it should be affordable from the global perspective if we look from the global scale. Of course, any and all such data should be open access, because otherwise it would defeat the purpose, really. Now, here I would add something that is not on this presentation. So when I gave this exact talk at the roundtable, there was also a bit of a discussion afterwards. There I was reminded about data sets that I partly knew about, partly very new to me. For example, the VIDA database that holds a lot of genotypes for cattle and sheep, or the SIMRI dataset that has really a wide range of chicken genotypes. Also, I did videos on various livestock and companion animal datasets from various species and sources. So I'm not saying that there is no data around. What I'm saying is that especially for the small breeds, there are gaps that could be filled. What I have in mind is some really easy system that would allow us to get any data within literally 10 minutes after thinking about it. So we think about any population, oh, I need data for this. I go to this place and I download it. Right now, if you think about Hereford cattle or Boer goats or something like that, you are in luck. But if you think about some of the many other local populations, you will probably have a hard time to find the genotypes for these. Also, another contribution to this discussion at this roundtable was that, which is actually true, that there is a lot of data around, but organizing them in a way that they are searchable and findable takes perhaps even more effort than getting new genotypes into the system. So, I mean, yeah, it's really challenging. The whole thing is really challenging. And that's why I'm bringing it up right now. So what would such a complete and open access database solve? Well, obviously it would provide an inventory of genotypes, but also give a complete overview about genomic diversity in breeds for particular species. We would increase our understanding about the livestock species, and also it would provide reference to determine genetic distances between them. This might be partly problematic, especially when it comes to transboundary breeds and small breeds, because some of these breeds, especially in Europe, are receiving subsidies. And of course, if it turns out that there is a larger population than expected, for example, in two neighboring countries, then some of the populations might not be categorized as endangered anymore, which is, well, a good thing. But then farmers might lose the subsidies for these, for keeping these individuals or keeping these breeds, which of course is a bad thing from their perspective. So these are in general also tough questions, but I also feel that we need to face these questions at some point. And then other undoubted advantage would be that such a database would provide reference populations in case we would have questions about purebred crossbred status. Because of course, if we want to do any kind of admixture analysis, we need purebred animals. And if we would have reference sets in such data set or database, it would be, of course, great. And of course, such a data set with widely available data from all of these small breeds of all the species, well, livestock species in this case, would really support the research of small and indigenous breeds in Europe, Africa, Asia, Americas, and Australia, and really all around the world. Now, after I unveiled my grand plan, let's see what can we expect in the future. Of course, I'm no fortune teller, so this is just my opinion. So let me know in the comments below if you agree or disagree with it. Now, what I know for sure that one of the central kind of marketing statements of small breeds or people who are working in safekeeping on small breeds is something that is stated in the quotation marks. So this is not an exact quote, but just the main message that comes up quite frequently. So this message is along these lines that it is worthwhile to keep these small breeds because in the future, a time might come when the specific characteristic trait or genomic variant of this small breed might be beneficial. We should keep these small breeds now, so large breeds or ultimately the mankind can benefit from them in the future. Now, I don't want to sound too cheesy, but I believe the future is now. And this is in particular with connection to the genome editing or gene editing, if you will. So genome editing is this new technique where you could actually cut the target location on any genome and add the modified piece into it. And then we have a new genomic sequence that incorporates this new gene. Now you might say that this is how you create a genetically modified organism or a GMO, but you are only partly right. Because in fact, the genome editing uses genes from the very same species and many times even from the very same breed. So the source genome and the target genome belong to the very same species and the very same breed. This also means that we could achieve the very same result with perfectly natural mating, but we need much more time. So basically what we gain with genome editing is time, and we can do a very specific edit and change just one particular gene of our interest. We do this on a level of zygote and insert this zygote into a surrogate mother, where after a normal gestation, the mutant founders are born, and these have already incorporated this new gene in their genome and spread it in a perfectly natural way afterwards. Of course, the flagship operation in this genome editing is the hornless cattle, but since then there are many other applications worldwide. Of course, still the question remains if individuals created by genome editing are genetically modified organisms. So this is actually also a rather complicated question, and I would leave the answering of it perhaps for some other time. But here I just want to underline that exactly this question led to the situation that at least in Europe, the genome editing was not really accepted, or at least there were serious doubts when it came to research funding. Now I could tell that nowadays the genome editing is beginning to be accepted, but personally I don't like this phrase at all. My personal opinion is that anyone who starts a sentence with "it's beginning to be accepted" or "it's starting to improve" or any similar phrases just shows wishful thinking. Personally, I think you either can show that it is accepted and you just say then it is accepted, or you cannot show it and then it does not give a message of false hope. So instead of a doubtful statement about the acceptance of genome editing, I show a few current news articles instead, or at least the titles, so I can prove my point. So the time of recording of this video is November 2021. And so these news are relatively recent. So you see that all of them are from this year. Now I put out this article also because, well, at the beginning, especially in the European research project, there were these doubts about genome editing. But as you see, nowadays, or at least according to this article from February 2021, Horizon Europe is going to fund research on genome editing in agriculture. Another news article from April 2021, the EU calls for rethink of GMO rules for gene-edited crops. This also points towards acceptance of gene editing in livestock, but also in the wider agriculture. Actually, I think in plant breeding, the people will have an easier time with genome editing than in livestock. Another news article from September 2021. After leaving the EU, the UK set new rules on gene-edited crop research, and also another article also from September 2021 announcing that UK is set to approve gene-edited livestock and crops in major post-Brexit break with the EU policy, which of course also points towards increased research in the genome editing area. Well, this is not about livestock anymore, but I thought it is also interesting in this context. So the World Health Organization has released a document that discusses genome editing in humans. Yes, you heard right. This is actually a set of documents that discusses the framework for governance in human genome editing. And I actually made a few videos on this that you could also find on this channel, where I go through some of these documents and comment on them. And the last news article I want to highlight here is also from September 2021, is about an apparent new possibility to genotype embryos. We talk about human embryos and choose and pick the one that is somehow the best, or for some reason, the embryo of choice. This, of course, has wide-ranging ethical consequences or ethical questions attached to it, which I don't want to discuss here. And I don't know even if I'm the right person to discuss these ethical questions. So there are 2 points that I want to make with this input. The first one is that if we get to a situation when genome editing or even other genomic techniques are being implemented on the human population. I mean, I'm not saying that they are right now, but there are discussions about these techniques in humans. At that point, the same or very similar techniques are much easier to implement in livestock. And the second point I want to make here, that especially this news article about picking embryos came really as a shock to me, because it is exactly the plot of the 90s movie Gattaca. So if you haven't seen this movie, I really encourage you to do so. It's a great movie, but also it is kind of a chilling one, especially in the light of current developments. So this is already my last slide here, coming back to livestock and coming back to small breeds. Well, as I mentioned at the beginning of this talk, this is kind of an uncommon presentation. I don't have answers. What I have are a lot of questions, and I wanted to summarize these questions again here on this slide. So when it comes to small breeds, where do we stand? What is the status of these valuable traits or valuable alleles that the small breeds can offer? Which ones are these? How we can find them? How can we implement them? Is there any repository where we could find the genotypes or even sequences of these small breeds? Are there any market-ready offers from small breeds or breeding organizations that could be used eventually in a context of genome editing? How can the small breeds benefit from the current genomic revolution? A lot of questions, but not so many answers for now. If you have also more questions or perhaps answers or suggestions for any of the things that were said in this presentation, I'd love to hear them in the comment section below. Also, I would cut the presentation here. I certainly thank you for your time you spent on this presentation and on this talk. And if you have any ideas, just really put them in the comments below. So thank you again and have a very nice day.